Okay then, so good morning, good afternoon, and good evening to the Pasteur Network community. Happy New Year. I wish all the best for each member of our community as we embark on our another day of collaboration. Another year, sorry, not day. Day also, but also a year. Okay then, so thank you very much for joining us today. I'm Hisham, the coordinator of the monthly seminars. As always, we'll begin with our speaker, followed by a Q&A session. So please free to submit your questions in the chat or ask them directly during the Q&A. Please, a quick reminder to ensure a smooth experience. Keep your microphone and camera turned off during the seminar. And please note that the seminar will be recorded and will be shared with our community on our YouTube channel. Today's topic is Frugal Science, a toolmaker approach for tackling problems in global health, ecological monitoring, and science education. And for that, we are very pleased to have with us our speaker, Manu Prakash. Manu Prakash's lab use interdisciplinary approaches, including theory and experiments to understand how computation is embodied in biological manner. Examples include cognition in single-cell proteins and morphological computing in animals with no neurons and origin of complex behavior in multicellular systems. Broadly, his team invent new tools for studying non-model organisms with significant focus on life in the ocean, addressing fundamental questions such as how do cells sense pressure or gravity. Finally, his team dedicated toward inventing and distributing frugal science tools to democratize access to science, diagnostics of deadly diseases like malaria, and convening global citizen science communities to tackle planetary scale environmental challenges such as mosquito surveillance or plankton surveillance by citizen sailors mapping the ocean in the age of Octopi. So I'm happy to now to leave the floor to our speaker, and I will come back for the Q&A session. Thank you very much. The floor is yours, Manu. You can share your screen. I'll stop sharing my screen.
Okay, I want to just begin by saying what a delight it is to talk to everyone. I know there are many different time zones, and this might not be the most convenient time, but just I'm very happy. I just came back from Institute Pasteur in Dakar a few months back. I was also at Institute Pasteur in Paris and met some of the folks associated with the network. I would say in terms of the community, I've already spent time at Institute Pasteur Madagascar, significant time in Dakar, uh, and plans of spending some time in Cambodia. And I think one thing that I really value in thinking about a lot of tools that I'll share today is the value of sharing them broadly with the community and exploring what others can do with the sets of tools that we've been building. So I'm going to begin by saying the purpose of this talk really is to integrate and enhance collaborations. There's a lot of platforms that we've been developing, and I would be very excited if any of you find value and usage for the general purpose tools that we make and to kind of begin and start those sets of conversations. Uh, I'll talk about this theme that I often call frugal science, which is the idea of how do you really build high quality diagnostics and research grade tools for low cost so that we can actually distribute and scale and really build capacity in science. Um, I have a lab at Stanford. I teach in the Department of Bioengineering. I am on the board of Pivot. And then there are two startups that I've started since to scale up some of the tools itself. So there's a broad group of students involved and many, many, many collaborators that I'll mention along the way in development of some of these tools. Uh, I think before I begin, I just want to mention the philosophical framework for me personally is driven by I teach this class called Frugal Science, and many people come together from around the world, and then we try to tackle difficult sets of problems collectively. And I remember there was a student from Sudan a couple years ago, And he taught me this saying, kind of just the philosophical framework for what he said. There is an Arabic saying in Sudan from long time ago, don't create a pen that doesn't eradicate ignorance and shame. And here the pen is a symbol of technology. And I think sometimes I often think about this, that the role of technology and technical capabilities really have to be at the service of what we are trying to achieve. And often we lose that track. And so this is really the core message for me personally, is really thinking hard about how do we really point our technical capabilities to some of the hardest problems that we need to solve collectively as a society. I'm going to talk about three very broad problems. One of them is really around ecological surveillance. Many diseases that we all work on have hidden roots in ecological changes, and really the capacity to even measure what's happening in ecosystems. Now, that's very difficult because, of course, ecosystems are much larger, and sometimes we don't even know what to measure. And the cost of measuring changes in ecosystems, for example, mosquito and mosquito surveillance broadly, and what's happening in populations, explodes pretty quickly. So, this is one theme that we will explore in the end of the talk. Uh, the other framework I want to mention, I don't know how much time I will have to talk about this, but I want to the thread around science and science education is critical in bringing communities to engage in the sets of solutions. Many programs that we build have citizen science at the core of it. Many a times, tools and medical solutions, their adoptions really require deep engagement with the community. And then, in general, if you think about, we have 2 billion kids on our planet, and roughly a billion or more of them have absolutely no access to the experience of science. So I'll spend a little bit of a time, the tools that we've been building in sharing that experience broadly. And just, I love working with kids, and it's a phenomenal approach in terms of starting to really think hard about how do we bring everybody into the fold of life science. And finally, I'll talk quite a lot about diagnostics, and I'll begin with this, but just, uh, we have a changing world, and it's been fascinating to see that as we have been building sets of tools around most of the diseases that many of you care about, TB, malaria, schistosomiasis, these diseases are also changing along in our populations. And to a certain extent, this realization that we need to leapfrog our tools and new tools need to be developed. Otherwise, we would actually be, we're falling behind significantly in most of the goals. Malaria being an interesting example of what's really happening with the Anopheles stephensi and new locations where we are starting to see urban malaria rise. So unless we can integrate and really bring new tools to the table, it gets harder and harder to tackle many of the common problems that we've all worked for very long periods of time in new ways. So, I started in a very depressing way. I told you that we have environmental challenges, we have health challenges, we have educational challenges, and that might seem like kind of a doomsday scenario. And one thing that we've been thinking about is this is where the power of network is incredibly important. Most of these things are grand challenges, and these grand challenges require us to think really globally. And this is really something that is so beautiful about the Pasteur network and other networks that have sprung, is how do we really build a truly collaborative environment that a solution from one place or an idea from one place can be implemented in another place, and collectively tackling these problems, because neither do we have enough scientists nor the capacity to even observe what's happening on our planet in context of diseases, to be able to tackle something like this. So I think this is really where the network approach comes in into play. And I want to share a historic example. So we've been doing this in this framework of frugal science, which I'll talk about, which is the perspective of building tools and openly sharing them broadly with groups of individuals, communities. Uh, and historically, there is a really interesting example in astronomy, which started this network approach. So on the right, you can see there is this picture of Sputnik. It was the very first satellite that was ever launched. And at that time, we did not have capacity to see the sky and where the satellites are. And a very famous astronomer, Fred Whipple, started a network program. It was called Operation Moonwatch. And the perspective of the program was, could you give literally $10 telescopes? This was happening, in the space race and the Cold War, to communities around the world. So you see there Philippines, South Africa, United States, all across the world. They had started building this global network of citizens looking at the sky, monitoring, and being able to tell every new satellite that was launched. This was the only way. And this community driven program led to a phenomenal framework in astronomy of citizens engaging and participating and strengthening the capacity of astronomy. Now, this happened long time ago, and there is no reason why we cannot share scientific tools broadly to really be able to bring these tools to people. And actually, when I think about healthcare workers, I think about them as really one of the world's largest citizen science programs. So a lot of things that I'll talk about now are really driven by what can we provide to healthcare workers that are operating and working in field conditions, primary health centers, what sets of tools are necessary, and what can we build in the research capacity framework so that we can accelerate some of this science, but really driven by deployment of these tools to broader sets of communities. Uh, I think I've spent a lot of time in the field, as I mentioned before, and many of the ideas that you will hear about today all originated from the field. So once a while, I'll actually mention some of the origin stories, but there are infinite number of collaborators to thank along the way because many of these conversations lead to completely new questions that we try to tackle. Uh, so to begin, I think I'll share a few sets of tools. I don't think I'll have time to talk about all the tools, but everything that I talk about is accessible in publications, websites, tools that we build along the way. I think I'll try to touch key points for each one of these frameworks that I mentioned, starting with diagnostics and general purpose microscopy for a second. But the portfolio is accessible. If you go to prakashlab.stanford.edu, you can find every one of these sets of projects and sets of details. You can reach out to me. I don't think I'll have time to talk about every one of them, but there's more than a dozen projects that we have launched globally in this space, and it's valuable to actually spend some time, look at it on the site. Um, so let's begin with microscopy. I am assuming most of you use microscopes in some way or form in your science. And it's interesting, I was asking ChatGPT recently for what are the 5 most important tools for science, and microscopy came second. Although I think mathematics came fifth, I would have thought mathematics should be number one. But still, it's important to note that microscopy plays a very critical role in thinking about science. But ironically, if you really ask, are microscopes accessible? I mean, A, they really don't operate in field conditions. So utilizing microscopes in field conditions is very difficult. And secondly, most of these automated platforms that become accessible are all black boxes. They're incredibly expensive. And many of the new applications and new protocols and AI that is developing so rapidly in microscopy is completely inaccessible to majority of institutions across the world. And so one of the focus that the first start that I want to really talk about is rethinking microscopy itself. Uh, there is just so much incredible beauty in the microscopic world, and there are incredible answers, and especially now with the age of being able to combine microscopy and genomics in what is being called spatial biology, which I'll talk a little bit about. We have an incredible opportunity to tackle new pathways, new diseases, and be able to see many of the perspectives of the diseases that we all care about, not just in lab settings, but if these microscopes can also come out of the lab and spend time in the field, you really get to see the kind of patient perspective that's useful. And I'll give you a few examples of that. Over the years, we have built many different kinds of microscopes. I'll talk about Foldscopes in the education space for a lot of ecological monitoring, planktoscopes. Uh, but majority of the time, I want to spend on automated instruments and microscopes that we have been building. And they are called, I love marine organisms, so we have named them Octopi and Squid. Octopi is really designed for diagnostics microscopy, that you could do AI-driven microscopy in a primary health center, and I'll show you examples of TB and malaria. But also what we realize is that we can build an open platform for highly complex microscopes, including confocal, completely automated robotics, to automate many of the assays that many of you might be working on. For the first time, bringing low cost, high throughput microscopy to communities around the world, and especially research institutes around the world. So this is the portfolio of microscopes. There is of the order of 25 or 30 instruments that we have built. They're all autonomous. They run on open source software, and so anybody can engage and build on top of these. And I'll give you examples of a few of these, although I won't have time to talk about every one of them. So let's just start with microscopy for a second and malaria for a second. I've had the fortune of spending a lot of time with my collaborators in Institute Pasteur, Dakar, and especially Mukhtar, and spending time with him in clinical settings, spending some time in partners in Tororo in Uganda, and some amount of time in Ranamapan in Madagascar, sort of made us realize the ground reality of malaria diagnostics, for example, and especially very low parasitemia and asymptomatic cases. And there's lots of challenges in malaria diagnostics. The first one is this is one of our field sites in India. And if any of you have done malaria microscopy, this is what it feels like. This is a one-room primary health center in Kalahandi. And Prem and Durga here are working to diagnose patients whose slides have come. They will spend of the order of 10, 12 hours every day doing microscopy. Hundreds of patients show up, and it's very, very difficult to upkeep. And one of the challenges has been is human-based microscopy has had a very hard time keeping up with especially lower parasitemia. The assays that we use currently in microscopy in malaria were really invented in 1904. The Gimsa stain came with the first set of the paper. So none of that has actually changed, which is quite dramatic if you think about almost a century has passed. There are other challenges in the current frame of thinking about malaria, and I'm using malaria as an example, and you'll see I'll broaden this platform out very quickly for other diseases. Number of diseases and the number of patients are starting to rise. We have HRP23 gene deletions, so many of the RDTs are right now struggling, and we also have treatment challenges associated with it. Simultaneously, we have new vaccine candidates coming, and that requires us to expand our capacity to really be able to also see asymptomatic cases. So the first question we really asked over many years is we have self-driving cars running around. I live in San Francisco, and I see literally right outside my street these self-driving cars pass by. And every single time I am my jaw drops seeing that, oh, there is no driver in the car. And almost 10 years ago, we asked this question, why can't we really do this for microscopy? This is a bigger goal. Lots of people have been thinking about it, but we took an approach of building it in the open and building open source instruments that really everybody could engage and build upon. And that led to the program called Octopi. These are modules of Octopi. It has imaging modules, it has robotic modules, it has computation, and each one of these things comes together to build what is an Octopi. You can see starting from 2016, uh, all the way on the very top, the sets of instruments were 3D printed, and now almost 10 years later, we have really built products that we can deploy in the field, very robust tools. And this is one example of Octopi, but I'll just show you kind of what goes inside this tool. This is a robotic instrument, and I'll just switch to kind of show you what it feels like. So I'm about to show you a video, and what you will see is completely hands-free, autonomous diagnostics of malaria. These are malaria slides. This is a cell phone video taken but it's kind of fun. And as I play this video, you'll watch that every single second the screen is changing. That's roughly between 10 to 15,000 blood cells that the instrument is analyzing in real time. And in real time, it's running a machine learning model to actually do the inference to say which cell is a parasite or not. So the amount of time it takes for you to move the slide from one place to the other, the instrument has actually captured all the cells, segmented them, run a machine learning model, and actually given you an answer. So as these things are flickering right in front of you, it's picking up kind of a needle in a haystack. Octopi is little like a little box. It's running on a gaming laptop right now. And this is actually data collected with partners at Institute Pasteur in Dakar. And we've been deploying many of these instruments across in Liberia, Cameroon, lots of partners in Uganda, Madagascar, and India. And one of the power of this tool is explore what we can really do with this, and especially both in hands of researchers, but also in hands of community health workers. So the way the assay works, you can take a slide, whatever you put on the slide, it uses a traditional glass slide, so there are no fancy cartridges, there are no supply chains. You put the slide in, and within 2 to 5 minutes, the instrument will image and analyze between 2 to 20 million cells. Now, you might be interested in immune responses, you might be interested in other sets of pathogens, and we build training models. These are convolutional neural networks that then are trained and in real time with no network connectivity. So this is not telemedicine in any way. The instruments require no internet and they operate on a battery. So for 8 to 10 hours, the instrument can operate without electricity, really collecting the kind of granularity of data. So we first targeted this on malaria, and to just show you the kind of data sets, this is a data set from Tanzania from one of our collaborators. This is field collected data, and you can see the instrument in an autonomous manner collected these massive fields of view, analyzed them, and really provide sets of insights both for research and for clinicians. And one of the power of this as a tool is, and the reason we are able to do this is we are actually not just doing microscopy. What we are implementing in this tool is spectroscopy. And what this means in simple terms is that the tool is measuring not just the morphology of cells, but it's measuring the DNA to RNA ratio in a cell. We use a dye called DAPI, and when DAPI binds to DNA versus RNA, it introduces a spectral shift. So we can measure the ratio of this DNA to RNA to give rise to these incredible data sets. And this is really why the machine is so fast. So I'll give you just another preview of the machine of what this looks like. Now you can see everything is happening inside this box, but I've removed the cover so you can actually see what's happening internally. And this is not sped up. This is a real-time video of an instrument operating. It's now autofocused and it's starting to detect parasites. And one thing we were excited about is when we built this, we started sharing this with communities around the world. We started building a network and we stumbled upon a problem. And this is kind of also the power of sharing the tool and realizing sets of problems. We were collecting data sets in India, and most of our smears look like this. And any of you who has made a blood smear knows this is not a good sign. And we started looking what is going on. And then we realized that many of the community health workers were actually making slides in the field. And, this was the conditions that they were making the slides in. And it occurred to us, and we realized that for the last 100 years, we've all been making smears, but we haven't really thought about what is the physics of making a smear. So I'm going to take you back to your lab days. If I'm curious how many of you have ever made a blood smear, you all know you take a slide, you put it on, and you just push it. Now you have to push it with the right velocity and the right angle, and often enough, the smear qualities are not good. And literally using a 70, we sort of asked ourselves a question, what is the physics of making a smear? So here are the equations. Nobody had mathematically done this. When we did this, we realized we can now build a small little gadget made out of simple parts and, tens of cents of parts that really allow us to automate smear making. And I'll kind of just show you what that looks like. So this object that I'm about to show you has a syringe inside. We use this syringe as a constant velocity motor, and that led to this project called Inkwell. Inkwell is a 3D printed smear making device. It has no batteries, no electricity, just a spring, a syringe, and a valve from a bicycle to regulate the speed. And you'll see the device is able to make perfect smears every single time. We have tested this for tens of thousands of cycles, and it's using the mathematical equation that I had described before for an optimal angle and optimal velocity. And now we can make smears with literally 20 million cells on a slide every single time. That brought standardization to our program, and it's actually very interesting. We've been able to share this with people, and this is a completely open source tool. Anybody who wants one, we actually ship them to people to bring standardization. And that allowed us to then start collecting large scale data sets. And I'm not going to spend too much time on the malaria data set, but one of the things that we have demonstrated with this is the first study that's coming out from our work in Uganda, that for 25 parasites per microliter, the machine is capable of doing 100% sensitivity and specificity, and coming down to 5 parasites per microliter, the machine can do 95% sensitivity and specificity. Now, these tools, the power of machine learning is that they change and improve over time. And so what we have been doing with Institute Pasteur Dakar is really mapping and increasing our capacity to collect large, broader sets of data sets. And anybody who's interested, these are the sets of partner sites that we have been working with. And any of you who are interested to join in the program, one of the things is deploying the instruments is pretty straightforward. And we have been starting to build a network for really building the largest digital database for malaria microscopy. And it's been really phenomenal to actually share this with community health workers and students across the network. We've been teaching courses. This was a course in Dakar on diagnostics. And one of the frameworks around this is that we've been able to essentially build a broad community of people that are sharing things like that. My Papa is on a call. I have my daughter here for a second. Just give me 1 second. Can I see it? Okay, but Papa is on a call and you have to go to school. Okay? Yeah, I'm sorry, but I'll talk to you when you come back. I'm gonna stay home today. My daughter wants to share that she's built a bus. Okay.
Whoa.
Go. Papa is gonna run. Yeah, go to school. Sorry about that interruption.
No
I hope
worries, no
you
worries.
enjoyed, and I'm sure in all your lives you have wonderful interruptions like this. This is morning time. I'm supposed to take the kids to school, but today they'll go by themselves. Okay, so
Yeah.
I was talking about malaria. One of the things that happens with these sets of tools also is massive amount of data is collected. And so we've been looking at chatbots and many of these AI-driven tools, and I'll just give you this example that we recently built to really access these data sets and explore many kinds of clinical insights that exist in these data sets. We've also been building clinician-friendly tools, which are zero code, so you can literally chat to the microscope. You can write to it, you can ask, could you analyze the data this way? And this chatbot is starting to write code to really just analyze this data set and explore it with you in real time. And so the power of this is the fact that now finally not only we have the capacity to do automated microscopy, so for around $10,000, we can build Octopi as a network, but literally the kinds of thousands of patient data sets that these can build, you now have the capacity to interact with these data sets to find clinical insights. Now, these tools are in development, but one of the exciting parts is we started sharing this tool and the community started asking for newer and newer function. The first one we built on it is why can't we train the tool to detect gametocytes? And we decided to then train it. So now we have the capacity to detect gametocytes. Very recently, we started a collaboration with Institute Pasteur Paris, where they wanted us to really tailor the tool for vaccine development. So we're starting to build assays to really be able to focus on both the species that are used in vaccine development, but also critically able to measure sets of parameters that are needed in those sets of assays. So you should not think of this just as a diagnostic tool in a primary health setting. It is also a tool in a research setting. Collaborators in Nepal reached out to us. They were interested in sickle cell anemia. And we looked at it as like, why can't we retrain the model to really do sickle cell anemia? And suddenly, with sets of partners at UBC and in Nepal, we demonstrated that we can do all four types of sickling on a platform like this. Now, what's very interesting about this, as you start to see the trend, the machine remains the same. So the model is that if these AI-driven models can start growing, the same instrument can run hundreds of assays. And the analogy here is a little bit like the App Store for diagnostics. So if any of you use smartphones, you know that there are hundreds of apps written by many developers. The hardware remains the same, but you can start building different apps. And this is the vision that we've been building. Recently, we started working on TB and demonstrated that we can do the similar framework for detecting live TB. And this is published work that shows that we can essentially do the same type of quantitative work that I showed with malaria with TB. I am looking for partners for work in TB. So if any of you who work in TB, I would be really delighted to implement a program, and especially because we have not started working on the clinical side of TB in this framework. Uh, Same goes for sexually transmitted diseases. And so, with these collaborators, this is a group in Kenya, we have been deploying these tools and engaging them in training them, also in the process of building the machine learning models, because it's very important to essentially have clinicians be engaged in training and annotating these sets of data sets. Now, these microscopes, as I said, are not just microscopes, they also do spectroscopy. So very recently, we implemented a low-cost open source Raman microscope. This is something that I'm very excited about because Raman microscopy can cost $150,000 for roughly around $8,000 in parts. We were able to show that we can implement a Raman microscope on this platform. And we have demonstrated this for two very specific applications. One of the application has been really in sepsis, to be able to identify label-free using Raman spectra on this microscope, identification of bacteria in individual bacterial species in blood samples. And then the second thing, which is actually quite surprising, and these are portable microscopes. So as you can see, this is Baba on the left. This was a paper that just came out in PNAS. We can demonstrate that with this portable instrument, we can do label-free identification of many of the antibiotics resistance, something that really can take a long amount of time from a drug susceptibility testing. We can take an assay that takes weeks and implement that in a minute. And that's happening because the molecules on the surfaces of these sets of organisms are starting to change, and Raman is very, very sensitive to surface molecules. So this is another platform now. We modified this platform to also do multiplex ELISA. So you are starting to see a trend, and I'm going to kind of start going exponentially faster because I still want to share many, many tools. Uh, so the framework really is that many of these tools are very, very flexible and they are adaptable to many sets of applications. And it took us 10 years to make them very robust, but now we are very excited broadly deploying them and broadly sharing them with the research community to build new applications on top of it, very much like the App Store, and the App Store both for diagnostics and research. Now, one of the interesting aspects of this is when we started building these tools, we also started realizing that a little bit of robotics and expanding them allows them to then compete with the research instruments that are out there. So up till now, I mentioned to you Octopi, which is on the left, which is this portable, smaller scale instrument. And we decided to start building instruments that can also replace almost any kind of microscopy needs, but now done in an open source framework, including confocal microscopes. So this is now Squid. Octopi, we often use the word for clinical microscopes, and Squid are for the research microscopes. And one of the power of this is really taking this modular approach, teaching people how to build these instruments, building many applications on top of it. And then suddenly, if a new assay is developed by Institute Pasteur in Cameroon, that can immediately be adopted by someone in Paris or the other way around. And that power of network is really incredible in sharing the sets of instruments. So this framework involves sharing instruments, sharing applications, sharing training, and building a larger community around this. So we have started building small labs in partner institutions where everybody has similar instrumentation. And I'm going to show you the few instruments now. So this is the Squid framework. Literally any instrument that you can imagine, we have a capacity to build in this framework. And we have started to build this with hardware, software, and community. All software is completely open. So it allows you to get under the hood and essentially start using the tools. And I'm going to give you a few examples of the types of applications, including drug screening. We do a lot of spatial transcriptomics and spatial biology, CODEX assays, and many of the RAMAN sets of things that I gave you. So this is the first example from Liberia. We were interested in CODEX assays because with NIH as partners, they've been studying Ebola survivors. And many of these survivors have incredible immune system because they did not get any drugs, no treatment, but they fought Ebola. And to study that immune system, you need to understand what is happening with their PBMCs and many sets of cells in their immune system. Prior to this, all the data and the samples were collected in Liberia, and then they would be analyzed in labs elsewhere. But we wanted to bring these assays, and especially an open source CODEX assay. And so with Dave and John and many sets of partners in Liberia, we implemented now a fluidic engine. Now this fluidic engine talks to the microscope and runs the kinds of assays that suddenly leads to 25 color CODEX assay implemented. So you can choose any of your antibodies of interest, and we can label them in a stepwise fashion, and you can really characterize the patient populations, but in a field setting. Similarly, on an example of thinking about long-term imaging, this is really an example from aging to watch organisms live in incubators. People started asking about implementing these things. I know many of you probably do live microscopy. So now it's the same Squid platform implementing in a live incubator. And many of these modules have been growing for a while. So this is now the example of a Squid with a confocal microscope to really increase the resolution. One of the applications that a group in Uppsala reached out to us about was, could you do low-cost, high-content screening? And this required robotics. So what you can see on the right is the Squid microscope, and on the left is an open source robot arm. And I'm going to give you an example. And now 5 of them are operating for months, collecting these incredible data sets. So these are cell painting assays where you can study the phenotype of the cell. You can introduce the sets of drugs that you're interested in. And now at a very low cost, even a small institution can run high content screening. So to just give you an example, this instruments that you saw literally runs for 3 to 4 months continuously. This is run in a 96 well plate assay, and then this is the kind of resolution that you have on these 96 well plates. The entire process is robotic, and this really is kind of one of the frameworks that There is a lot of drudgery in life science, and one of the things that we're starting to do is starting to eliminate some of that. Similarly goes for optical pool screening. There are many CRISPR screens that are now accessible and available, and we are building a completely open platform with fluidics, robotics, and microscopy combined together to do these types of assays. So one of the ways that you should think about optical pool screening is essentially in situ genomics, that you can do a phenotype by observing cells by imaging them, and then transition and actually do a readout that's associated with sequencing, but without removing the cells from the platform, so you actually know what phenotype matches to what sets of genotypes. This is example of the Squid confocal microscopes, and kind of, one of the beauty of many of these sets of tools is the fact that they allow you to do volumetric imaging, they allow you to really see. And one thing that I'm really excited about is people building completely new kinds of applications on top of it. I'm going to show you one framework of a video and then switch to the molecular side of the story, and then probably only take 10 more minutes. So the big picture vision that I want to share with you is microscopes are kind of like robots. And what we have to do is to build these robotic farms, and what we are calling them, which is a common framework that's now being used, which is self-driving labs. Could we build low-cost, open frameworks that you can really program your complex assays, and the machines can run to give you large-scale data sets to then explore? So this is one example of this integration. So if you watch this video, there is an incubator in the back, which is storing many sets of well plates. And again, all of this is integrated by these open source tools and hardware. We've been building all of this hardware. This is an open source robotic arm. We've built these sets of plate readers, and now there is on the left is the Squid, and you'll start see that it'll start imaging. It'll pick up the plate. Once your imaging is done, it'll put it back in the incubator, and the process continues. And one of the power of these sets of tools is that once you can do something like that, you can imagine the kinds of assays that you could have never run previously, and also never run in a small scale setting, because many of the big pharma companies have incredible amount of automation. And what we need to move towards, and one of the big goals of the network and of the community for us, is to really make all of these sets of things broadly accessible. So we've been building this network and sharing these sets of tools. And one of the things that we recently did is also started a manufacturing arm, which is called Cephla, that now allows us to deploy these sets of tools with sets of community partners. So lots and lots of tools and community. The network is growing very quickly. So these are the research sets of partners. And what I would be very excited about is starting to build a framework at Institut Pasteur, where many different Institut Pasteurs have similar sets of instrumentation, and they can share sets of assays. So I already know I'm running out of time, so let me give you a very brief preview on the educational framework and very brief preview on the ecological side, and then hopefully we can take 10 minutes for questions. So on the education front, I think I am assuming some of you might know, but one of the passion for us that we have is how do we bring all of this sophisticated instrumentation that I showed you literally in classrooms around the world? How do we bring it even in our communities? So, best researchers are the ones that are passionate about science, and we have to engage with communities early. So almost a decade ago, in around 2011, with my student Jim Cebulski, we decided, could we really make a microscope? The microscopes that I showed you before still cost around $5,000 to $10,000, $15,000. But what if you could make a microscope for $1, $1 to $2? And we asked ourselves a question: could you make a microscope that costs $1 to $2 and can see single bacteria? So that led to Foldscope, which is a paper microscope. It's a single sheet of paper. Everything is printed, including the optics. You fold it together, and you have a microscope that can image single bacteria. And I'm just going to show you this video to give a sense that these sets of instruments and microscopes don't require any electricity. They don't require cell phones. If you have a cell phone, you can connect And now this is not a magnifying glass. This literally competes with something that would cost $2,000 to $3,000 because this has a capacity to image 1 micron as a resolution. And one of the power of this as a tool is because it's connected to cell phones, now you can bring microscopy literally anywhere. And there's lots of clinical applications people have been implementing. There's lots of applications in animal health, plant health, but also in education. So this is one example I wanted to show you. You can see these ciliates and single bacteria. So all of these videos that you are seeing here are collected with this $2 paper microscope called Foldscope. Now, one of the contexts of this is really what happens when you share this. So we decided to share this tool broadly. Roughly around 3 million kids around the world use this tool now. This is a picture from India, Bay Area. This is Lebanon. And one of the kind of contexts is that there are, again, AI layers that are built on cell phones, but the primary goal for this is to really bring science to the masses. And what does it happen? And so this is one of the communities currently, and many of the institutes that we work with, ICGEB South Africa is a great example. They work with us as an institution, and then they deploy these microscopes very broadly in their community to really bring science outside the lab. And that's incredibly important. So many of these are pictures from around the world. And one of the power of this tool is that there is a community platform where everybody shares their data openly. So you have an idea, you share it, and all of these data sets are documented on this community platform called Microcosmos. And this allows people in the community to talk to each other, and not just scientists talking to scientists. These are scientists talking to kids, talking to retired people. Again, going back to the image that I showed you about astronomy, can we do that in a community scale? So a mosquito entomologist sharing a picture of Foldscope collected data, and out somewhere else in a lab, somebody else seeing that data. Those sets of connections are critical and very important. We've been building national programs around this. So this is a program in India. This was roughly around 500,000 kids. There's lots of online tools that are accessible and hundreds of publications of community engaged work associated with different sets of fields in agriculture, public health, lots of machine learning tools and diagnostics built on top of it. So this is called Foldscope. It's very easy for us to implement this in community sets of partners. And then maybe in the last 1 minute remaining, I'll just mention that everything that I told you about microscopy, we actually also do for molecular diagnostics. So the threads around building molecular tools, and the last tool that I want to share is it's called SnapDx, which is really about now could we do a molecular assay in the middle of nowhere using literally no electricity. So this is a current paper that we'll be publishing soon. I realize that I'm out of time, so I'm going to stop here. But the thing that I really want to mention at the end of the day is if you have interest in using these sets of tools, reach out, because what it allows us to do is to build a broader community around these sets of platforms. And what I'm excited about is enabling for you to take many of these tools in directions that we cannot even imagine. I'm going to stop sharing my screen. I'm hoping we can still have a few minutes for questions. And I am here and delighted to engage.
Thank you very much, Manu. It was quite impressive. Amazing tools, and I think many people from the Pasteur network would be interested by the collaboration with you and using your tools. So we opened the Q&A session, which we'll try to have like 4 or 5 questions max. So we have 15 minutes, if you don't mind.
Yes, and then I just shared my email in the chat, so please reach out. And if you just write Institute Pasteur Network on the top, I'll kind of poke that up on the top.
Yeah. So we will share it when I will share you the video of the seminar to the whole community. I will share also Manu's email address. So who has a question? for the moment.
And you can really ask anything. I mean, you can also ask about things that I didn't talk about. This is really an informal conversation. Noel, go
Yes,
ahead.
please. Noel.
It's a tradition. I'm usually asking the first
Yes,
question
it
because no
is.
one wants
Thank
to
you,
ask
Noel.
questions here. So really, that's really interesting what you present, and I'm interested in two parts. We are working in close connection with community. So indeed, I'm the director of the Pasteur Institute in Guinea. And you saw that in Guinea, probably one of the main wording for making the future is Simandou. And Simandou is a big mining, the very young mining situation on which the country is trying to base his future. This means that in forests that are usually protected forests, we are in a wet zone where you are like in the Ivory Coast, you can have a lot of destruction in order to make the train going across and railway station and etc. And so we are working with villages in which we are looking at what this modification of the environment is making in terms of modification of rodents, micromammals, bats, and the risk of transmission. And one of our main difficulty, and we are making a lot of effort on that, I could share with you some of the movies we have done. We found that movies were the best way to understand, to make the people understanding, is that we are going from the field to the lab. But it's very difficult for the people to understand what is a lab. And what you are proposing in terms of a small microscope at $2, allowing to show something to the people, even if it is not really what we are working on, would be really, really very interesting. So I don't know in which way it will be possible.
I mean,
So
I think I would be very
one of my
excited.
questions. This is one of my questions. And the second question is for the other machines that are more sophisticated, let's say. One of the problems we have here also is connection and the way of transmitting data. If you have to organize by AI the data in distance. So how are you solving this? I'm stopping here.
So
Thank you.
two quick things. So first of all, I am just delighted that you brought this up because I think one of the goals is, and I didn't get a chance to talk about the molecular tools. There are other sets of assays where we have built that allow us to do both molecular mosquito surveillance for new diseases. Similarly, being able to build a molecular assay with no electricity. So what I showed you about microscopy, there are other layers to it. I think on the community side, it is literally very straightforward because we have built a community of 3 million users around the world, and many of them are organizations like yours that implement programs, and then the organizations also talk to each other. So we have a very similar example of deforestation in Brazil and Peru, where we just came back from the Amazon and massive amount of gold mining that happens, sets of communities that live there and change. And I think some of this is really driven about sharing the tools and letting the community decide what types of programs and how. So this is also conversational. This is a phenomenal example. And I think what is beautiful in the example that you shared is that there is a direct link between what's happening in the community and what should happen in the research lab, because you might find sets of new things that you need to be then working on. So if you just send me a message or I'll write to you, straightforward for us to implement. Tens of thousands of Foldscopes are very easy to bring out, implement programs, and have people explore and build educational content on top of it. Species discovery is a big thing. There are hundreds of species, microbial species, that have been discovered using Foldscopes. So that by itself gives credit to the community. The second part, on the network side of the story, the power of many of the AI-driven tools is it depends on what data you store. In early phases of programs, we store everything. So when we digitize a slide, that's roughly around 5 gigabytes of data. That would not make sense to transmit. But because the machine is interpreting and analyzing it in real time, I'm only storing the pictures that where the parasite is. And it can be retrained in many ways to what is of interest, and what gets transmitted is really that, oh, I found this many sets of parasites, and locally you can store certain things. You can also store a minimum. So a normal slide with one single glass slide with 20 million cells is a massive amount of 5 gigabytes of data, but only the parasites is only a few kilobytes, even though highest quality images are collected. And then when you have analyzed it, only a few bytes carry the information of what total number of parasites were there. So there is a gradation associated with it. But what we are excited about digitizing many of these assays is because they become incredibly quantitative. So one of the challenges is with and without automation, when you run something like this manually, in half an hour with 100x field of view, you can see something of the order of 5,000, 10,000 cells. While with a very simple automation like this, you can see somewhere of the order 20 million cells. So it starts to change the granularity of what you can ask a question for.
Thank you. Thank you very much.
Yeah, and then if you could please send me some of those videos, I would love to see this, because there is a complete parallel between what you said and what we are doing in the Amazon.
Okay, I will do that this afternoon.
Yes. Yes.
I think those videos are online, Noel, no? On your
Yes, they're online, but I will put the
Yeah, I
connection.
think this way I'll have your email and other threads.
Yeah,
Okay,
yeah,
Sung,
I will send you an email. I will send you an email.
yes, Sung has a question.
Yeah, go ahead, Songpin.
Go ahead, Sung.
I think you're muted.
Okay. Oh, no. Am I
Oh.
on now?
Yes, yes.
Yeah,
Okay,
go
good.
ahead. Thanks.
Yeah, I raised my hand to say hello to you all. I'm the CEO of IP Korea. It's 1 o'clock. I
Welcome.
really appreciate Manu for the wonderful talk. And I'd like to mention one thing. We have been working on TB
Mhm.
for quite a long time, and we have high throughput screening system and the HCS system. So if you could, assist us, then, we may build up a better system for the high throughput screening.
Yeah.
So I just want to mention that, I may contact you to
Yes.
discuss
Yeah, and I think
about how.
we are looking for TB partners, because what we're realizing, I am slightly biased towards malaria just because of my background, but it's important that many of these tools have direct implications in both on the diagnostic side, but also on the discovery side. So yeah, I think it would be fantastic, and especially if you've been thinking about this as a problem. We have done work in TB, but not to the level of extent that I would like.
All right. Thank you very much. So it's very nice to talk to you.
And I didn't even know
Thank
that
you.
there was a IP in Korea, so that by itself is good news for me.
So we have a 20-year of history of IP in
Wow.
Korea.
Wow.
And thank you very much for being here, because it's quite late for you. It's 1 o'clock, 1 a.m.
Yeah, it's
in
1
Korea.
o'clock. Yes.
So,
Yes.
thank you.
Still, I enjoyed your talk. Yeah.
Thank you.
Thanks a lot.
Any other questions?
Yes, please. We still have some time for 1 or 2 questions. Well, many people already left, because I think it's quite late for
Yeah, no,
many
I
of them.
can
But
imagine.
yeah,
Yeah. Yeah, I think maybe if we don't have a question, one thing I wanted to mention. Oh,
you have a
Renard has a
question.
question.
Oh no, please. And please, could you please introduce
Hi.
yourself? Yeah.
Of course, Hisham, like always, I'm Renaud Vatrinet.
Like
Actually,
always,
I
thank
think
you.
that we've met Manu, I think it was 2 years ago at IP Dakar. So now I'm at the Pasteur Network Association working in Paris, and I'm a scientific and education officer. It's not just a question, it was just to mention to you, and as a reminder for the Pasteur Network community as well, is that we are going to have a working group now on specifically dedicated to diagnostics, among other thematics. So I think that you can be a very good contact when once it's in place, because it's all about networking, it's about partnership. So once it's in place, I think we'll think about you, and I think that you could bring some contribution to it.
Yeah, no,
So
I think
thank
I would
you.
be very happy. With diagnostics, you have to understand the markets very well. You have to understand use cases very well, and they're very different country to country. So building them in isolation makes no sense. And it's fantastic diagnostic technologies are built and then they fall flat because they really did not have the context of those specific markets in place. So for us, it's very important. And I mean, in the Octopi side, the thing that what we've been doing in Senegal has really been very valuable because it tells us at the grassroots what is needed and it pushes the technology. And so
Excellent.
this would be phenomenal if there was a working group. And specifically when the tools are just starting to work, we are very excited to share it broadly because the number of validation and usability studies that can be done in different contexts explodes. And in parallel, many of the people that engage get to completely learn on a new technology. And often enough, most diagnostics are platform technologies. And so I think the big focus that we have right now is to build a framework where platform technologies are shared and other people can build layers on top of it to contextualize it to the diseases of highest importance. So that requires partnerships, that requires IP agreements. It's actually very valuable to have some kind of a working group because that gives a relationship that's very different. So yeah,
Excellent.
if you'd have anything written on this working group, just send that to me. I would be very happy to take a look.
Thank you.
Sorry, I was muted. Thank you very much for your question, Renaud. We'll take a last question. Yeah, I think we're good. I don't know if Sankid, you still have a question because you're still raising your hand, but I don't think so. I don't think so. So,
Yeah,
Manu,
and I
I
think
have,
I already mentioned my email is manup@stanford.edu. The lab website is just called prakashlab.stanford.edu. And if you just send me a note, and then I'm happy to also just send a brief set of links to people that engage. So if they need links to read more about it, read many of the papers, I can tally and put them together in one place, and then you can share it.
yeah, definitely. And actually, I'm taking also this opportunity because we are now on one of the WHO Hive platforms. So I will share it to the whole community where we can discuss and exchange in a secure way.
Yeah,
At
and
the
I think
same
what
place.
I would be excited about is I don't know whether there is a technical get together that happens for the network, because that way in one single location, I can meet many people in the network and we can bring many of the tools just so that people can play with them, kind of get a sense and think about what they might want to do with it. It's kind of an interesting framework. And then I think the thing we did not discuss much is really around teaching. One of the important things about these tools is these tools don't come out of nowhere. They come from a very rigorous framework of uh, learning, and that invention framework can be taught. So I teach this class, Frugal Science, but then I've also been thinking about designing classes very specifically. And if the Institute Pasteur Network, especially for students and postdocs, if they're really interested in learning the framework of invention in many of these things, uh, usually it's valuable for this type of a course to be offered to much more of a network. So if you've thought about the education side of it, that's also another very relevant scenario. Because for collaborations, what's valuable is when students from many different institutions work together.
Totally. And actually, so I was
Oh,
about to
Kathleen
talk
finally raised her hand.
Kathleen. So thank you very much, Kathleen.
Yeah.
No, I just wanted to say that you have a course at IP Dakar, right? That
Yes.
is a very successful one, and I would urge people from the network to go and see when you are having these courses, and urge them to be part of that. And that's really
Yeah, I think that's what I've been thinking about, is
fantastic.
whether to teach them in person or whether to teach them in isolation or kind of online. And yeah, I think this is something that I'm really excited about. I really feel strongly that at the end of the day, teaching is really the mechanism to really translate many of these tools.
Completely agree.
Yeah. So Kathleen and Renaud will be your your contact person about trainings. If you want to develop some courses or training within the network. So yeah. Okay. So thank you very much, Manu. Thank you very much for your time.
Thank you so
and
much.
this
Thank you for folks that
fantastic webinar.
stayed awake this late, and I'm available, happy to chat.
Yeah. Thank you so much, and see you next month for the next monthly webinar.
Yeah. Bye,
Bye-bye.
everyone.
Bye. Bye.
Bye,
Ciao.
everyone. Bye.
Bye-bye.
Thank you. Bye-bye.
Thank you, bye.
Bye-bye.